everyone. I'm very excited to have this opportunity to present at the virtual ACA and to tell you all a little bit about some of the things that we're doing at the Crystallization Center at HWI to enable remote crystal growth screening and imaging. And a lot of what you're going to hear about in today's session has a lot to do with remote data access. So your sample is ready and you want to collect data on it. What we offer at the HWI Crystallization Center is remote access to crystallization screening, so a step before you have your crystal. And the reason for that is because in any crystal-based structural experiment, the first thing you actually have to do is find conditions in which a crystal will grow. Whether you're doing X-ray crystallography or microcrystal electron diffraction, and finding these conditions is still a major bottleneck. And so we offer remote access crystallization screening to make this a more efficient process. So who might be interested in making use of this remote access crystallization screening? Maybe you have already tried all of the different screens that are available in your lab. Maybe you manually set up all of your experimental trays, or you manually inspect each of your wells for crystal growth. Maybe you're an under-resourced institute or a minority serving institute. Maybe you don't have access to advanced robotics or imaging. It could be, given what's happening right now, that your lab isn't open or is minimally staffed because of COVID-19. Other reasons: you might want to efficiently and rapidly screen through 1,536 different conditions. You might want to be able to quickly distinguish protein crystals from salt crystals. You might want to detect submicron crystals. Maybe you're interested in doing XFELs or microcrystal electron diffraction. And you might be tired of actually having to manually score all of the images of your crystal growth, and you want to take advantage of being able to auto-score those images. So at the Crystallization Center at HWI, we do offer, or are soon to offer, all of these different options. And I'm going to tell you a little bit about that. But before we do, I want to address a question up front that I sometimes get, which is, what if you already know a condition that crystals will grow in for your particular sample? Is it worthwhile to do any additional screening? And it's a great question, and my answer is yes, I think it's always worthwhile to do additional screening. And why might that be? Because multiple crystal forms can often be helpful. So crystal packing can include active sites. Different experimental techniques can actually require different types of crystals. So if you're doing room temperature X-ray crystallography, you really need big, chunky crystals. If you're doing microcrystal electron diffraction, you need extremely tiny crystals. And so the type of experiment you're doing can dictate what type of crystal you actually need. And finally, I think that one of the really important things to keep in mind is that different structures can often provide a more complete picture for driving hypotheses about biomolecular function and the potential mechanisms of action. And so as structural biologists, it's important for us to kind of consider all of these different things. Okay, so the Crystallization Center at HWI, what we do is we offer a remote option for high throughput crystallization screening for your samples. So we have a particular microassay plate that has 1,536 different wells, and so we can screen very efficiently for 1,536 conditions in this one plate. This is equivalent to 16 96 weld trays in the same physical footprint as one 96 weld tray. So that enables us to really use a very small amount of protein sample and a very small amount of crystallization cocktail. We provide the robotics, imaging instruments, and expertise. So the way it works is that we use microbatch under oil. And so we have oil, we have our crystallization cocktail, and we also have the protein sample. And so we use a robust imaging schedule to monitor for crystal growth. We always start with before a protein is added, and then we monitor over the course of 6 weeks. In this particular well, we see that a protein crystal appears after 4 weeks. So this can be really wonderful because it really enables you to see very quickly whether crystals are forming within particular conditions. And one of the questions that we can ask is, well, what if the bright field images are actually ambiguous? So if we were doing this live and in person, I would ask for a show of hands and we'd do some audience participation about whether any of these particular wells actually contain protein crystals. But I'm pre-recording this, and so I'm just going to tell you at the outset that all 4 of these wells contain protein crystals, but each of these would be somewhat difficult to identify using just bright field images. And so at the Crystallization Center, we use a Rock Imager 1000 with Sonic, and that has second order nonlinear imaging of chiral crystals. And so what this means is that it's got a femtosecond laser that actually enables 2 different methods: ultraviolet 2-photon excited fluorescence, and UV TPF essentially enables excitation of aromatic residues that will then fluoresce when they're concentrated. So concentrated can mean that it's crystalline. It can also mean that it's precipitated or in a phase transition and kind of blobby. And then second harmonic generation, which is a way to identify chiral crystals. And so most crystal forms actually will generate a signal here. And so you can use the combination of these 2 techniques to be able to very quickly identify crystals, and that those crystals actually contain protein. And so it's a way for us to identify or distinguish between salt crystals and protein crystals, as well as to be able to identify when proteins are just concentrated but not crystalline, and when they actually are crystalline. And so this is an example that you will actually see again later. So for all of the experiments that we screen at the Crystallization Center, we make use of the Rock Imager 1000 with Sonic. And this again enables us to really quickly verify protein crystals and to detect submicron crystals. And I'll show you some examples of that in a second. So we image everything. So this is actually one of the experimental plates that is 1536 conditions, and with bright field UV TPF and SHG. And we are, I think, important to mention is that we're the only facility that actually offers this type of imaging to external users. And so that's really exciting for us because it really enables some things that I think would be really difficult to do without this type of imaging, without these types of imaging experiments. So how can you make use of that? Well, using these types of images helps really easily identify protein crystals. So in this particular case, it's very easy to see these big crystals in even the bright field. What's interesting is that we actually have 2 crystal morphologies in this particular well, which again you can very easily see with using all 3 imaging modalities. Sometimes your well is just filled with precipitate, and I know that I have often encountered wells that look like this, and what's great about using the UV TPF and SHG is that it enables you to actually detect when the precipitate is actually obscuring actual crystals. And so this is, again, really exciting. And for us, and for some of the research that I'm doing, which has to do with being able to use nanocrystals and submicron crystals, the ability to detect crystals that might otherwise be considered failures but are actually just really, really small is really, really exciting. So in this case, the bright field image really doesn't show any crystals, but if you look at the UV TPF and SHG, you can see that there are very nice, extremely small crystals. So again, this is really exciting. So one of the difficult things when you're doing high throughput crystallization screening is that you end up with a lot of images that you have to actually look through. So each of our 1536 experimental screens actually results in a minimum of almost 14,000 images. And so that's a lot of person hours to actually go through each of those images and be able to assess whether a crystal is forming or not. So the Crystallization Center, in collaboration with Google Brain, other crystallization centers around the world, and pharma companies actually developed an algorithm called machine recognition of crystallization outcomes. So it's called MARCO. And if you're interested in more details, the reference is here. It uses bright field images and has a large testing and training set that enables us to very efficiently and quickly rank crystallization outcomes as crystalline or other classifications. So we're currently working on a GUI interface that allows this to occur very quickly and easily. And we're also working on incorporating the UV, TPF, and SHG images, because right now these images only use, or this algorithm only uses bright field images. So I have a very talented student named Ethan who's currently working on the GUI interface. It's called Marco Polo. I'm going to show you a little video of how this works. Essentially, you can open up your images that you've obtained from the 1536 experimental screen, and you can set it up to classify. And what that will do is go through and classify as either precipitate, other, clear, or crystal. This is a subset that doesn't necessarily happen this quickly, but the crystals you can then rank by crystals according to how Marco has scored them. You can zoom in, you can tell, actually let's back up for just a second. You can tell in this case that those are crystals. And there are a lot of occasions where crystals are identified. We can verify that using the UV and the SHG. And we can zoom in, we can zoom out. We can actually even look at things, for instance, over time. So we're going to show you all the dates here. What you can see is at the beginning of the experiment there as a control before the protein is added, and that actually crystals in this particular well grow very quickly and this is classified as both crystals by Marco and by the human scoring. We have a Marco confidence that we get out of this as well. And so this is currently in beta testing. We're hoping to have this available for beta testing by users sometime this year. One of the tricky things that we experience is that the image sizes in terms of the number of pixels for the bright field UV, TPF, and SHG images are actually different, and this makes it so that one can't just overlay them very easily. So some of the additional things that we're doing to be able to start incorporating UV, TPF, and SHG images into this pipeline are to use different imaging registration methods. So if we can take our 1224 pixels by 1024 pixels and then 512 by 512 pixels, and we can actually register them so that they're mapped to the same grid, same coordinate system. That then enables us to do pixel level machine learning algorithms and do wavelet transforms on these images. Using the images in this way will actually enable us to, for instance, do kind of multi-scale analysis, to do threshold detection, to detect particles of particular size, and to develop ways to do nanocrystal detection that are currently really not available. And so these are some of the exciting things that we're actively working on right now in the Crystallization Center. Some of the other things that we are working on incorporating, or will soon be working on, are moving from not just doing 1536 experiments, but also to take the positive experiments there and do optimization using the same conditions that we used to make the 1536 experiments. Also to do in situ crystallization, to be able to do essentially a screen to beam type of technique. We're really excited because we've got a cryo-EM center going in, and it will be opening sometime this year. And our first microscope has a C to D detector, and because we're really uniquely suited or uniquely poised to detect the very small crystals that one requires for microcrystal electron diffraction. We're very excited about this. And then eventually we're going to be moving to doing, to enabling some high throughput drug screening with these small crystals that we can detect. So I just want to leave you with a couple of success stories for some of the users who have sent us samples. This is actually a particular structure of a transporter that is important in a particular disease. And again, bright field UV, TPF, and SHG shows these very small but positive crystal hits. that were the initial conditions that went on to enable solving this particular crystal structure. And that was from the Thibodeau lab at the University of Pittsburgh. This is from the Snell lab. Tim Stokowski is a soon-to-be graduated PhD student from Roswell Park Comprehensive Cancer Center, and he might recognize these two images from earlier. These particular, again, in this bright field image, you might not pick out this as a crystal, but this is the condition that ended up being the initial crystallization hit that was optimized to enable Tim to solve this structure. And finally, a very recent story, a very recent success story is the work. We've been screening a lot of SARS-CoV-2 related proteins for crystallization, and a recent experiment that we set up for some scientists at Oak Ridge National Lab on the main protease from SARS-CoV-2 actually yielded thin plate-like crystal flowers that appeared within a week. They were able to optimize these conditions very quickly and solve at room temperature the X-ray crystal structure of the main protease of SARS-CoV-2. And so this is just recently out in Nature Communication. We're really excited to be doing a lot of work in SARS-CoV-2 right now. So with that, I will actually wrap this up and say thank you for your attention. Contact me if you're interested in finding out about how we do crystal screening, or are interested in sending some samples. And again, thank you for your attention.